So we're going to start with this session with a presentation by Dr. Anastasia Henry. She's the Senior Director and Staff Scientist at Denali Therapeutics. Her research deals with how to target the lysosomal system in Parkinson's, including the recent clinical trial of a LRRK2 inhibitor. So yeah, Anastasia?
Thank you, and thanks so much for the organizers for the opportunity to update on some of our work for LRRK2. So just taking a step back, I know some of this you probably heard a little bit in terms of the genetics around LRRK2, but I'll do a little bit as it really through the lens of how this guides our therapeutic strategy as we think about trying to target LRRK2 for PD. So if you look at the genetic landscape for PD, you see this convergence on genes that function in the lysosomal system, and these are highlighted in green here. As you can see, LRRK2 in red kind of is across the spectrum where there's mutations that have a stronger effect in terms of disease risk, as well as more common risk variants that lead to a slight increase for developing PD. So there's genetic support for this as a target. This was identified, about 20 years ago in terms of the first pedigree analysis that really started to identify this association between variants in LRRK2 and PD. And it's one of the most common risk factors for Parkinson's disease. So as you think about really having a strong genetic rationale, this supports that this is relevant for PD, but it doesn't tell us how we could potentially target this therapeutically. So when you look at the protein structure for LRRK2, it's a very large multi-domain protein. Most of the mutations center around the active part of this protein, the kinase activity, the GTPase domain that do its business. And when you look at these mutations that are associated with increased risk for PD, these seem to converge on leading to increased activity of the kinase. So I won't go into all of the cell biology, but it was identified about 10 years ago that these LRRK2 phosphorylates these RAB proteins, which are essentially master regulators of trafficking in your cell. And so when these get too much phosphorylation by LRRK2, your trafficking doesn't function properly, and this can lead to defects in the lysosome, which is essentially the cell's garbage disposal system. And so when you look at the mutations that are associated with PD, these, we and others have shown that this leads to significant increase in kinase activity as read out through phosphorylation of LRRK2 substrates. So these tell us that for the variants that are associated with decreased risk, you have increased kinase activity. There's also information from the genetic associations that support the idea that reducing LRRK2 kinase activity could be beneficial. So there's this dual variant, the N551K R1398H variant within LRRK2, which is associated with decreased risk for developing PD. So a few years ago, we sought out to try and understand what this variant was doing to see if this could help guide and inform our therapeutic strategy for targeting LRRK2. And what we did here was we took cells and had them express this variant and then assess what are the consequences for LRRK2 activity. We saw that this led to a reduction in the phosphorylation of LRRK2. So mutations in LRRK2 that are associated with increased risk for PD. There's also increased kinase activity, and mutations that are associated with reduced risk for PD seem to decrease the activity of the kinase. We were also able to get human PBMC samples from carriers of this variant, or those that did not carry, and we also saw that in those samples we saw a decrease in LRRK2 activity as read out looking at phospho-RAP10 levels. So if you kind of think of it as a spectrum and how this could guide our therapeutic approach, too much kinase activity increases disease risk, and variants that are associated with reduced seem to decrease risk, providing therapeutic rationale for LRRK2 kinase inhibition. So we began developing LRRK2 kinase inhibitors when Denali started, essentially, and I'll talk through this. We've pursued 2 different LRRK2 kinase inhibitors in our early clinical studies. First, we confirmed that these led to dose-dependent reduction in LRRK2 activity. I'm showing data here from human cells, either PBMCs that were isolated or cells that we differentiated into microglia to understand what our compounds would be doing on relevant cells in the brain. And we saw a dose-dependent reduction in LRRK2 activity, looking at either phosphorylation of LRRK2 itself or one of the proteins that it phosphorylates. So this confirmed that we had, compounds that were doing what we want them to. They're inhibiting LRRK2 effectively. So as we think about, I talked about the genetic association, but how is it related to pathways that we think might be important in terms of Parkinson's disease? Well, as I started my talk, there is this, appreciation that the function of the lysosome might be compromised and impacted as we age, and that this can be exacerbated and lead to accumulation of different proteins, including alpha-synuclein. So there's many different approaches that people are taking to try and restore the function of the lysosome to get it to be able to degrade proteins normally, including clearing alpha-synuclein. LRRK2 is one of these proteins, so it functions in regulating the lysosome. It localizes to lysosomes when they're stressed. And our idea here is that if you have too much kinase activity, this can inhibit the function of the lysosomes, and we've shown this, and I'll go through some of that data, and that when you can inhibit this, you can then restore lysosome function. You reduce the Rab phosphorylation, and you can now get proper activity, proper ability to degrade things. So to explore this, we first started in cellular models, and so I'm not sure if you can see here, but these were cells we took from mice, where we're expressing either the G2019S, one of the common variants in LRRK2, or we've deleted LRRK2. And this is looking at just what do the lysosomes look like in these cells. And so normally on the left in a wild type mouse, you see many different spots, but in the middle, the G2019S cell, I think you can appreciate you have this alteration in the morphology that correlates with reduction in function of the lysosome. In the LRRK2 knockout cells, we were able to see that you actually see more lysosomes and function is increased. So when we treated with one of our LRRK2 inhibitors in this system, we're able to rescue that defect in the lysosome morphology, and this correlates with changes in function. And so this is a complicated slide, but essentially we're using an assay to look at the ability of lysosomes to degrade proteins. It's an assay we can use on microscopes. When you have reduced function of the lysosome, you have reduced fluorescent signal. And so we looked at some of our cell models. We're expressing a different variant in LRRK2 associated with PD, the R1441G variant. We saw a reduction in lysosomal function, looking at protein turnover. When we treat with our lead clinical molecule, DNL151, we're able to completely restore lysosomal function. So this got us excited in the ability of LRRK2 kinase inhibition as a potential strategy to not only reduce kinase activity, but potentially restore lysosomal dysfunction. So an update on some of our clinical studies. So I'll give an update of where we are and a little bit of data on some of our past studies as well. So as I mentioned, we first started looking at two different kinase inhibitors, DNL201 and DNL151. We conducted extensive testing in both healthy volunteers as well as human subjects living with Parkinson's disease. And I'll go through some of the data, but in using samples from subjects from our clinical trial, we were able to confirm that we were inhibiting LRRK2 and that it's safe and tolerable. And based on the dosing frequency, we chose DNL151 to move into late stage studies. With 201, we'd have to dose multiple times a day. 151 would be a pill once a day, which is better. So we are looking at we're working on this program in collaboration with Biogen, and we are currently in two late stage studies. We have our LUMA study, which is in subjects with sporadic Parkinson's disease, as well as a biomarker study that we just started at the end of last year, specifically looking in carriers of the LRRK2 mutation. And the idea here is, there is compelling data for the mutations that leads to increased kinase activity, but there's also a lot of data suggesting that in sporadic PD patients, there's also an increase in LRRK2 activity. So trying to test the therapeutic hypothesis of whether this could be beneficial not only in LRRK2 mutation carriers, but also idiopathic Parkinson's disease patients. In terms of some of the endpoints that we're looking at, in addition to some of the clinical scales, we're also confirming, are we inhibiting LRRK2? I mentioned the lysosomal pathway and that being associated with Parkinson's disease. We're trying to use readouts of lysosomal activity to understand if we're correcting lysosomal dysfunction, as well as PD relevant endpoints looking at things like alpha-synuclein, NFL, which is a marker of neuron loss, and then DAT-scan. So here's some data from our early phase 1 studies with DNL151. So first we wanted to see, are we actually inhibiting LRRK2 in our studies? And as you can see here, we're using either a readout of LRRK2 phosphorylation itself or of one of its Rab substrates. And we saw a nice dose-dependent reduction in terms of LRRK2 activity looking at both readouts. This is in blood samples or in PBMCs that we isolate from blood from subjects in our study. So this confirmed that we're inhibiting LRRK2 effectively, which is great. That's the first step to then test the therapeutic hypothesis and see if this can be beneficial. We also have promising data to suggest that we're improving lysosomal function. So one great readout for us for lysosomal function in our clinical studies is this lipid called BMP. I'm not going to go into all the biology around it, but essentially it accumulates when lysosomal function is not working properly. And we know this from studies in rare diseases that affect the lysosome. You see BMP levels are elevated. And so we see a dose-dependent reduction in BMP levels when we look at biofluids. Here I'm showing data from urine from subjects dosed with our LRRK2 inhibitor. So this suggested that we are having some improvements in lysosomal function at the doses used in our clinical study. So I mentioned LUMA. So this is our study that's ongoing in sporadic Parkinson's disease patients. So as you know, as a design, we're not specifically enriching here for LRRK2 variant carriers. It is focused on subjects that are early, within 2 years of their diagnosis. And as we think about primary endpoints, it is really trying to look at more of those clinical endpoints. I showed some of the primary biomarkers in terms of target engagement, but really trying to understand what are the effects of this on some endpoints that are relevant for disease. So an update on the trials. We don't have any data yet, unfortunately, but we will next year. So it's fully enrolled. We have 650 participants that have engaged in this study across 13 different countries, many sites, and we expect the data readout in mid next year. So definitely excited to see the data from that study. Nothing to report now, but stay tuned. As I mentioned, we also late last year started an additional study. It's a phase 2A study specifically in subjects that carry a variant in LRRK2. And the idea behind this, so I didn't go into this, before. We actually had a study in LRRK2 variant carriers in collaboration with Biogen. It was a different study, really trying to look at beneficial effects in terms of some of these clinical endpoints. Based on what we knew and the data we had, it could have been a very, very long study. And so in collaboration with them, the decision was made to not move that study forward and not continue that. But we really believe in the therapeutic hypothesis that, if you have a LRRK2 mutation, you have increased kinase activity, and that would be an ideal patient population that might benefit from LRRK2 inhibitors. So we started our own biomarker study, really trying to understand specifically in patients that carry a LRRK2 mutation, what are some of the effects in terms of some of these biomarkers I talked about, and can we understand more around what LRRK2 inhibitors are doing in subjects that carry a LRRK2 mutation? So the study is ongoing, and I think this will be a really nice sister data set to have with the sporadic Parkinson's disease patient data to really understand what sort of biomarkers are changing. And it'll be very helpful to interpret the data because we don't know. It will all benefit, patients benefit from a LRRK2 inhibitor, will be subsets, and I think this study can help guide what to look for and what sort of pathway endpoints we can see that might be improved. So stay tuned, it's actively enrolling in the United States and in the EU, and we're really excited to see how some of this data rolls out. I went through that pretty quickly. That's so I can pause here and take questions.
I have one. The second study, the BEACON study, you were talking about treating more as a chronic condition then, as opposed to treating what might happen because of the genetic makeup, as opposed to what has happened. Am I interpreting that correctly?
Yeah, so both of the studies are in people that already have Parkinson's disease. This one, it would be really thinking of people that carry a mutation in LRRK2. So I think it's more around the biological hypothesis of LRRK2 hyperactivity. It's very compelling in people that carry a LRRK2 mutation. We know those are associated with increased kinase activity. So that's a great, ideal group to understand whether this could be beneficial. So we also hope there's a lot of potential in sporadic Parkinson's disease, and that is just, there's less data, right? So we wanted to look at both of the populations because even if it's not beneficial broadly across sporadic PD patients, if it looks good in this study, that could be useful as we think about subsets potentially of sporadic Parkinson's disease patients that might benefit from a LRRK2 inhibitor. So we're hoping these are kind of sister studies that could help inform who might most benefit from LRRK2 inhibition because we're still in our early days of understanding its potential.
Patients have you enrolled so far in BEACON, and what is your target?
Yeah, I don't know if that I can publicly disclose that, sorry. And I just can defer, I'm the cell biologist, so I don't know. But I don't think we should that publicly, sorry. But it's in the early days, it just started this summer.
What fraction of PD patients have this LRRK2 mutation?
Yeah, I think it varies widely depending on the area of the world you're talking about. So I think there's estimates, 1 to 5% of PD patients might carry a LRRK2 mutation. We don't know, there's likely also subsets within that don't, that have increased LRRK2 activity through other mechanisms. We know that There's other genetic variants that increase PD risk that also seem to increase LRRK2 activity. And we've also done a lot of work to show that when the lysosome is compromised through other genetic variants or stressors or toxins that you're exposed to, that also increases LRRK2 activity. So there's a defined genetic population, which is small but sizable. We also think that there are likely people that don't carry LRRK2 mutation, but their activity is elevated through lysosomal dysfunction, exposure to toxins or other things. And I think that's what we're trying to understand more about. A, can we get a measure of whose activity is elevated? And then it'll be really interesting as we get our data to understand which people might benefit the most from a LRRK2 inhibitor, and can we correlate that with things in terms of elevations of LRRK2 activity or changes in some of those markers of lysosomal function. We'll be looking at all those lysosomal biomarkers to understand how that kind of all correlates together with clinically meaningful readouts. And so we don't know yet, it's a proportion, but there's also other people we think might benefit that don't carry a LRRK2 mutation. But our study is kind of the most advanced, so we'll be kind of the first to really understand this and see this current data.
And how about trying to go about finding participants? Because you need to do that deep phenotyping. You're going to, and I'm not sure that clinical records as they stand at the moment are doing enough of that deep phenotyping to help you with these quite complicated studies. What can we ask the clinical community to do to support?
I think that's a great question. I know we've been working with a lot of different groups to really find LRRK2 variant carriers for BEACON, right? And so anything that we can do to help people understand their genetic status so that people have that information, and with clinicians so that they know we have this study, so if they are sequencing people. But I think, you know, knowing that the study exists, hopefully that helps to get more excitement and inspires more people to do that analysis, because there's an option for people then to enter into a clinical study if they do have these mutations. So I don't know if that's the answer in terms of what we can do, but it definitely would be helpful, anything that we can do to identify more people with LRRK2 mutations, that, because it's slow, right? And I think this is taking longer just because it is hard to identify all the LRRK2 variant carriers.
I know you're working with Movement Disorder Society and WPC and all of those groups to try and enable some
Yes,
of that.
yes, yeah, exactly.
Oh, Caitlin's got a question.
I just have a question about just LRRK2 inhibitors in general. Presumably LRRK2 has some sort of role in the cell that's positive. So does inhibiting it, you know, stop it completely? How do you kind of strike a balance, I guess, between, you know, eliminating it altogether and keeping it at a functioning level, I guess?
I think that's a really excellent question. And so I think, you know, from our assessment from others, if you completely get rid of LRRK2, like there's no protein present, I don't think that's a good thing. You start to see alterations in lysosome-related organelles. When we've looked at assessment of lysosomal function, completely getting rid of LRRK2, you actually do have some deficits in lysosomal function. We don't see those with LRRK2 inhibition, where you still have LRRK2 present, it's just its activity is reduced. So I think there are some differences about whether you completely get rid of LRRK2 or keep it there. But you're absolutely right, it has a normal function, right? We don't have this protein for no reason. We're starting to learn more about what that normal function is. Over the last 5 years, I think we've learned a lot more that it does seem to be sensing and responding to lysosomal damage. It could be doing a good thing at certain points. Our hypothesis is that it might actually be helping to correct lysosomal damage, but if it gets stuck on over your lifetime and you continue to get damage just through normal aging or exposure to toxins, it might not be as effective at responding to new damage. And so that over time that starts to be an issue. So I think that is a question of do you affect any of those early lysosomal damage responses? We don't have any data to suggest it's an issue, and in our current studies, there's no safety issues that we've observed. But I think that's something we'll have to continue to monitor just to understand, are there certain contexts where you don't want to inhibit LRRK2? I don't think you want to get rid of LRRK2, and I think it is important here to note that we're not fully inhibiting LRRK2 as well. We're reducing it, there is still some activity. From our data in cells, it suggests you need to inhibit it by about 80% to have beneficial effects in LRRK2, so that's kind of where we're targeting. But I think it's kind of going hand in hand with a lot of the basic research that's going on to really understand what is the normal function of LRRK2. It was discovered, the link 20 years ago, but we still don't know a ton about its normal function.
Thank you very, very, very much indeed, Anastasia.
Thank you so much.